All right, so let's get started with worms. This is the planarian worm, Smithia mediterranea, which has it's a freshwater worm. It lives in fresh water, and it's about between 1 millimeter and 1 centimeter in length, and possess distinguishable anatomy and shape. You can see it has a couple of eyes, pharynx, and with these stainings, we can see their internal organs, including intestine, uh, muscle fibers for movement, axonal projections, a complex brain with two lobes that we have, nerve cords, etc. So despite this very complex morphology, we investigate this model organism because of their very, very powerful regenerative capabilities. So I took this worm and cut it the head. You can see that the piece, the head, regenerates in about 2 weeks all the organs that are missing, including a new tail in the correct position, new pharynx, and everything inside, right? The same worm, oh, here it is. The same worm, I cut it the tail, and after 2 weeks, it regenerates as well every organ that is missing in the correct locations. In this case, it's missing a head and a trunk, so it regenerates anteriorly a new head and a new trunk that is connected, innervating all the remaining tissues. You can basically cut any piece, almost any piece in the worm, and regenerates a complete organism. You can see as well that not only it regenerates the correct morphology, but also the proportions need to be regenerated properly. You can see here, let's see, is there a pointer? So you can see that the original piece is too wide compared to the length, right? But after 2 weeks, the worm gets elongated and the proportions become correct, right? So my lab tried to understand what are the regulatory mechanisms that allows these outstanding regenerative capabilities in terms of temporal and spatial patterns, and not only patterns but also forms. In this case, when we feed the worm grows and maintains more or less correct proportions. And when we stop feeding the worm during starvation, the worm can go backwards, can't degrow. To mention as well that the worm is asexual, can reproduce asexually by fission, is hermaphrodite, so can reproduce as well sexually, no problem with sex, and is immortal. So basically, it's the pinnacle of evolution, right? So, how do we understand these very complex mechanisms in space, time, gene regulation, etc.? So in my lab, we follow a systems biology of growth and form, where we try to understand what are the mechanisms involving genes, signals, metabolism, and biophysics that together allows these outstanding developmental processes. We follow this reverse engineering approach where we start with functional experiments. We can knock down genes in the worm. I'm going to show you that. We can formalize those experiments into mathematical models that then through more dynamic modeling, we can try to recapitulate those behaviors. Sometimes, many times these behaviors are very complex. So we can either find parameters through machine learning, and I'm going to show you as well how can we use machine learning as well to derive the whole model de novo. Then we can create predictions and go back to the lab to validate them. And we apply this methodology to development, regeneration, synthetic bio, and human health. So let's start with development. So for the functional experiments, we have also developed new molecular tools that streamline gene cloning for the knockdown and the interrogation of the locations of the expression of different genes. So through these new methodologies, we can very rapidly clone genes and create these in situ expression patterns where we can see the locations of the RNA of different genes. In this case, this is a SLIT1, which is a gene that is expressed in the midline and is extremely important for the worm to differentiate the center versus site. Because you can see that when we knock down the SLIT1, instead of regenerating 2 eyes in a regenerating head, the worm regenerates a single eye. Why? Because it cannot distinguish center between side. So the only way to solve the location is to regenerate an eye in the center. Moreover, we can knock down genes that are essential for the specification of the tail. We knock down beta-catenin in this case and cut head and tail. The trunk is going to regenerate two heads, one anterior and one posterior. So this makes the planarian worm a very good model organisms with all these genetic tools. So how the worm regenerates their shapes and their forms. So we know that if we not knock down anything, right, a trunk piece is going to regenerate a head and a tail. But when we knock down, for example, beta-catenin, we can alter the organs that are going to be regenerated. In this case, now we have two heads. And again, the heads have brains, eyes, etc. We can also do long-term knockdowns. So here we are knocking down the worm for weeks and weeks without any amputations. It's the in homeostasis. So regularly the worm just grows, but here when we knock down the beta-catenin, again necessary for specifying the tail, the worm becomes round and has multiple eyes everywhere. So it seems that the worm has lost the ability to coordinate the anterior posterior with the mediolateral axis, and instead everything becomes a head in the external side and then something in the middle, right? So this gives us clues about the mechanisms coordinating this axis. Another important aspect is the mediolateral, as I showed you before. During the regeneration, a head amputation is going to produce two eyes, but when we knock down the slit one, the midline, it can produce a cyclop, a single eye in the middle, because the neoblasts, the stem cells, cannot differentiate center versus side. We can also do homeostasis. So if we knock down the slit one without any cuts, any amputations, the worm is going to start regenerating a new eye that is in the center, because there's a lot of turnover during homeostasis, where the stem cells replace all the cells in the body of the worm. So we have also performed RNA-seq. This is not the fancy spatial transcriptomics that we have seen yesterday. This is a shortcut where we can cut the worm into different pieces across the anterior-posterior axis. In this case, it's 6 pieces, and then do bulk RNA-seq in those pieces. So we can have pseudo-spatial transcriptomics. And you can see that there are genes that are expressed in the head in a gradient fashion, in the trunk, in the tail, and in the border. And these genes, we believe, are very important to specify not only the location of the morphological parts, but also the shape. What are then the regulatory mechanisms that can maintain and restore the patterning and whole body shapes? That's unknown, and that's what we are trying to understand. Specifically, first, how the head and the tail is restored. What is the mechanism that restores it? So for that, we went to the theory of Turing reaction diffusion that you are not familiar. It was proposed by Alan Turing in the 1950s. And it involves just two molecules, an activator that activates an inhibitor that inhibits the activator. Importantly, they can diffuse at different rates through tissue. So these self-regulated mechanisms, they don't need any other inputs, can produce these beautiful periodic patterns through time. So starting with random noise, it can produce these stripes patterns, and also not only develop them, but also regenerate. If I perturb the center of this reaction diffusion system, it can regenerate the missing locations. So now this doesn't look like a worm at all, right? But now let's focus on a smaller domain, a smaller piece in this reaction diffusion domain. We make zoom here, we can get something like this, right? Now we can see the valleys and the hills in this pattern. So the distance between the peaks in this pattern is what is called the wavelength, the Turing wavelength of the mechanism. And that can be altered according to the different parameters in the model. So something that Minhart proposed many years ago was if the wavelength of an organism is below the Turing wavelength, instead of having a periodic pattern, what we can have is just a peak in one side and a valley in the other. And that's what we believe is regulating the self-regenerating capabilities of the planarian world. Let me show you. So we have created these partial differential equations that capture these mechanisms where we can simulate head and tail patterns in 1D domains that when we cut them, it self-regenerate again the head versus the tail pattern. In this case, again, the worm length is lower than the Turing wavelength. So only one peak shows up in that domain. What happens when the domain gets longer? Because as I showed you before, the worm gets longer and longer. So what happens when the domain gets longer is that the system degenerates, right? Now more than one peak is going to show up, and not only that, but the head and the tail is going to become in the same colocalized locations, right? You see that instead of having head and a tail peaks, now the two peaks are going to be colocalized in the both sides and also in the center if it grows too much. So it's a problem, right? That doesn't follow what we observe biologically. So I can show you another phenotype that is very interesting in planarian, which is fission. So here is a single worm that was isolated in a petri dish, and in every picture, you can see that this was in the course of a few days. The worm produced these fissioned pieces by cutting itself. That's a method that the worm uses for asexual reproduction. Why do we isolate the worms? Because the population, when they are in the population, that's an inhibitor of the fission process. Is when they feel lonely, right? When they are alone by themselves, that the fission behavior triggers. So if I zoom into the first fission, uh, we can see very closely how the worm does this procedure, where it anchors the tail with some kind of biological glue, and then the head pushes forward until the piece breaks. Now, the piece doesn't break randomly in any location. We can observe that there are fission planes that are formed before this procedure happens. So how does these fission planes form? So we believe it's connected to the same genetic mechanisms that defines head versus tail. So when we have head versus tail, um, we saw originally that it's going to degenerate when there's no regulations between them. But now what we introduce is this cross-inhibited regulation. So you can see that the Turing system for the head and the Turing system for the tail now are cross-inhibiting each other. So what that means is that the model does not allow two peaks to be colocalized. So in that case, when the worm grows, is going to create these ectopic tail and head peaks that we believe is a trigger for the fission mechanisms. That's where the fission plane will develop. We can also introduce a gradient that controls the wavelength of the Turing mechanism. In that case, the wavelength, since it's controlled by a gradient, is going to be shorter posteriorly and longer anteriorly. And that's what makes the fission plane to appear posterior to the worm, as we see in the biology. We can change the parameter of this control strength to modulate the location of the fission plane from the center to more posteriorly. We can also modulate the strength of the gradient to modulate at which length the worm is going to fission, to calibrate the model to the experimental data, or to avoid it completely, as when we observe them in a very high density population, or maybe with low temperature or high light that inhibits the growth of the worm, the fission of the worm. So I show you how head and tail gradients can be self-regulated, can be explained by this Turing mechanism. But how about the worm shape? How the anterior, posterior, and the medial lateral axis get coordinated? So that's what we investigated next. First, we recorded the growth when fed and the degrowth when starved populations to see how the proportions and how the length and the width of the worm changes through time. So we use automated image analysis to extract these parameters, and this is how it looks like. So it's very linear. When fed and starved, the worms grow the length at a particular rate, and when they are starved, they change the length and width with the very same rate, but in the opposite direction. You can see in this panel, this is through time, and this is length versus width. These are the proportions of the worm, are slightly different of worms that are growing versus worms that are degrowing. And that change happens very rapidly. This is when fed, and then after 1 week, the proportions are already changing immediately. So it's a very rapid process. This is a very complicated phenotype in the sense that we have signals and we have as well cell behaviors, right? Growth and degrowth. So how can we model these type of phenotypes computationally? For this, we use continuous models. So these are partial differential equations where we have variables for cell density that is controlled by other signals. And also cell density gets, the cells get attached to each other through cellular adhesion proteins that can be also regulated dynamically. These are complicated models in the sense that not only we have space and time, but also we have non-local terms that define the adhesion strengths between two different cells depending on the cell adhesion molecules that they express. We have done some tests of these type of models, and here I'm showing you how a tissue made of two different type of cells, red and greens, expressing two different cell adhesion molecules, two different CAMs, can self-organize differently depending on the strength of adhesion between the two proteins. So when red and red are at the same strength and green and red versus green, everything is going to be a mix, right? When we simulate this, all the cells mix up. And now when they have addition molecules that adhere with different strengths, for example, here is the same between the same type of CAMs, but zero between the different type of CAMs. In this case, we can observe complete sorting of the cells. Another phenotype is when one of the CAMs adhere much more strongly than the other one, but there's still some addition strength between the different heterotypic CAMs. Then we can get these self-organizations where some of the cells get in the center and the other ones get in the periphery. This phenotype that has been observed is a classical phenotype of cell sorting that has been observed many times. So this is the type of modeling that we are going to apply to explain how the worm regenerates their shape or controls their shape. So how can we, how do we think that the worm controls this shape? How is the organization between anterior, posterior, and mediolateral genes happen? So we know that when we knock down no genes, then the worm regenerates from a head fragment, regenerates fine. When we knock down beta-catenin, which is an AP axis gene, the worm more or less has a normal shape, but regenerates to head. But this phenotype, when we are now knocking down both AP and ML axis genes, now we observe that the worm produces ectopic heads and loses the ability to maintain the shape. So the idea is that our model is going to include coordination between the AP and the ML axis, by which the stem cells, all neoblasts in the worm, are going to read that information, that positional information, to decide where to grow and where to go. So again, we have anterior, posterior axis genes like SFRP1, FC41. We have mediolateral genes, SLIT1 in the center, WIN5 on the sides, that are necessary to produce a worm. But how are they coordinating, right? How is that coordination happening? So for that, we then created a mechanistic model that can grow at least elongated shapes. So here the model includes AP axis genes, an organizer, This should be controlled by the Turing system, although here there's no Turing system, it's just over directly put there. We have AP-axis genes that are expressed and diffused through the anterior and posterior uh, most anterior and posterior cells. Then we have border cells that express border morphogen that is inhibited by those AP-axis genes. And then that everything controls the growth morphogen that controls basically the cell density, right? So again, these are continuous model of cell density where morphogens can diffuse within the tissue. We tried to calibrate the model by hand. That was very hard because the model has many parameters. So at least we proved that the model can grow elongated shapes, but these are very far, right, from the shapes that we observe experimentally in the worm. So how do we calibrate the model? So what we did was to create an intermediate descriptive model that can remove all that variability that we observe in the worms, right? The worms have different shapes that maybe the worm is looking to the right, to the left, etc. So what we did is to create this up-round shape, which is basically a rectangle with two caps, and calibrate very easily, right, by linear regression, growth model of the worm and at the growth model of the worm that captures the main shape of the worm during growth and degrowth, fed and starvation. So it is that the continuous descriptive model is a good approximation of the shape of the worm during growth and degrowth. Now we can use the model in an evolutionary algorithm approach, a machine learning approach, to calibrate the parameters of the model. So the idea is that we are going to have model parameters, the equations that I show you, that I didn't show you, but I show you the diagram at least, a mechanistic model that can be simulated, and then that mechanistic model can be compared through time to the descriptive model that we have of the worm. The algorithm works with an initial population of parameter sets that are random. Then we can evaluate the fitness in that way, select parents, crossover, mutation, and select the next generation according to how good the models are. The best models are kept in the population, and they combine themselves, they do mutations, they change the parameters to evaluate a new population again, and that process happens iteratively during many generations. One of the challenges of this project was that the simulation takes hours. The simulation of a single parameter set takes 3-4 hours to run because of the partial differential equations are very hard to solve. So the idea was, how can we optimize this machine learning methodology? So we restore to a process where we don't simulate the worm for the whole time, but we have a shape error threshold that dynamically changes as the models get better. So basically the fitness, how good the model is how long the simulation can proceed until it gets very bad, further than the threshold. So in that case, we don't need to simulate these models all the way, but only until they reach the shape threshold, the error threshold. Then when a model managed to simulate the whole 9 weeks of data, then we can reset the shape error threshold to to make the simulation more stringent and then simulate the models faster. If we see the generation, the evolutionary dynamics through time, you can see that it's very up and down in the sense that whenever a model manages to simulate the whole simulation, the threshold goes down and all the population error goes down, right? So that happens many times during evolution. And if you see here the base model error, it's getting better and better. These are 10 different runs, and it gets to zero, to very close to zero error all the time. So how does it like a model that has been calibrated? So now the model looks very, very close to what we observe experimentally. So this is the simulation, and this is the length, width, and proportions of the in vivo versus the calibrated in silico model, and shows how close the model is to the data. So this model is good at growth, and that's the only data that we use to calibrate the model, growth data. The next question was, can we use the model to figure out how the worm can degrow? What is the mechanism that transforms a growing worm into a degrowing worm? So what we did is a parameter scan. We knew that during the growth, the apoptosis rate goes higher. Why? Because the worm is not fed and still needs to replace cells that are dying. So the neoblast, the stem cell, are still dividing and replacing other cells. So the energy for that process comes from other cells. So the reason that apoptosis goes up, to give the energy to the stem cells to be able to differentiate. So we knew that the apoptosis rate should be higher, but we didn't know what other parameter was necessary because we tried only apoptosis rate and the proportions didn't follow the experimental data. So we tried any other parameter, diffusion constants, etc. And the only one that managed, these are all the simulations, the only ones that managed to recapitulate the growth proportions was changing the strength of the AP axis morphogens. So when we do that, when we change the pole strength and increase apoptosis, now the worm can recapitulate the degrowth behavior. That's a nice mechanistic hypothesis of how the worm is transformed between a growing to the degrowing phenotype. I showed you at the beginning, right, that the worm not only grows and degrows, but also regenerates. That's the next thing that we are trying to study. So here are time lapse of pieces of head, trunk, and tail, and how they regenerate and restore their proportions. Again, we do automatic image analysis method. We can now plot time versus length and time versus width. And you can see that there's two regimes. It seems that in the first 2 weeks, the worm restore their proportions, and then it goes back to the degrowth behavior. It's more clear if now we plot length versus width. These are the head, trunk, and tail. This is the size, the average size of the worms. We use worms of the same size. That's the reason that the average can be computed. And you can see very clearly that there are two regimes in these worms. One that is regenerative, where the proportions get restored, and even the worm grows during that process, even if the worm is not being starved, right? So it gets longer, it goes to the right, it gets longer, and it gets thinner. And after 2 weeks, and doesn't matter where do you start, after 2 weeks, the worm reaches the correct proportions and now continues the degrowth regime. Here. So we use the model again to predict what are the regulatory mechanisms that can control this process. Again, we did another parameter scan, and the only way that the model can predict regeneration is now with a high pole strength, higher pole strength here, and a high apoptosis through the body. That's how the model can recapitulate the regenerative dynamics that we observe. So in summary, with these modeling methods, we have hypothesized that there are 3 different regulation regimes in planarians. There's one that is a high pole strength and low apoptosis because the worm is being fed, and that's during growth. So during growth, high pole strength and low apoptosis can recapitulate these growth dynamics. When there's low pole strength and high apoptosis during starvation, the worm degrows. And finally, when there's a high pole strength and high apoptosis, that's during regeneration. In this regime, the worm can regenerate the correct shape. We are now trying to tinker with the signals to create other phenotypes and use the model to validate these phenotypes, and to use those experiments to validate further the model. All right. That's about development and regeneration, but in my lab, we are also interested in using this methodology for synthetic biology. So one thing that we have done in the past is to study metabolic networks in synthetic biology applications, how different bacteriums can degrade certain plant biomass. In this case, GLBIO is a metabolite that derives from cellulose. And we have created these dynamic models that can optimize the degradation pathways of this bacterium. I'm not going to talk about this, but that gives me the intro to show you Flaxer, just to advertise our bioinformatics tools. So Flaxer is a web application. You don't need to install anything to compute and interactively visualize genome scale metabolic models. So the problem with genome scale metabolic models is that they have all the reactions, can have all the reactions in an organism. You imagine thousands of metabolites and reactions. So if we build, if we when we visualize naively, right, like a force-based graph, it looks like a cloud of interaction that is impossible to see. So we developed is a methodology to create these very tidy graphs based on the flags in the network. We prioritize the reactions that have the highest flags during a simulation and draw a tree map, a tree graph of those reactions. And now we can observe the most important reactions first and then the rest. So this graph can be computed automatically in our server and then display in a very nice, interface. We can have different type of graphs. We can create pathways between two metabolites of interest, and this is how the diagram looks like. I mean, the web interface looks like. It's very interactive. You can zoom in, zoom out, export, etc. So you work with genome scale metabolic models. You can upload your model, or you can visualize one of the more than 1,000 models that we have uploaded into our server. You can upload your SBML model as well. So Flaxer, UMBC, EDU. Recently, we have expanded the methodology to include also the ability to merge, compare, and translate genome scale models. So one challenge in the field is that we have different metabolic and reaction identifier systems. Depending on the tools that you use to reconstruct these models, you can have incompatible identifiers. So what we did is to create a database that translates any identifier system, almost any identifier system, to a common metabolic and reaction universal ID. And now we can merge models, compare them, etc. So Mergem is a Python tool that you can just install with PyPy, but also we have included in the Flaxer website. So you can input, you can upload your models or use any of the 1,000 that we have there to create these very nice graphs where it shows you what is the commonalities between the models and what is different between the models, which is very useful for building new models to compare organisms, etc. We also encourage you to have a look at that if you work with genome scale metabolic models. Right. I said that we were interested in the synthetic biology. So what we are trying to do is to use all that we have learned in the planarian worm, how these gradients can inform the process of regeneration and pattern formation to try to engineer these processes synthetically. So the idea is that we have a field of cells. This square is a field of cells and two morphogens that are going to be diffusing from the orthogonal locations, the top and the left. So a field of cells, and the input is two morphogens that diffuse orthogonally. The idea is, can we automatically design gene regulatory mechanisms that can produce any pattern of interest, any synthetic pattern? So here is an example, it's the logo of the university. Can we create machine learning methods that can design gene regulatory networks de novo, gene regulatory mechanisms de novo, that when we simulate them, can develop the pattern of interest? Right. So first, for doing this, we did a framework that can simulate many types of gene regulatory interactions. So here we have single interactions, positive and negative, and then different ways to integrate more than one interaction. Can be necessary, a sufficient type of interaction, double negative, double positive and negative regulation. All type of combinations can be put together as building blocks to generate very generic gene regulatory mechanisms that can be simulated in the computer. Right. And in addition, we can have decay and diffusion. So now the challenge is that not only we are trying to derive the parameters of the model, we are actually building the model completely de novo. So we are building the equations de novo, or the computer needs to build the equations. So that's a much more complex problem. So we use high performance machine learning using the GPU computing. So the idea is the same type of method. It's an evolutionary computation where a set of models are evolving. Now it's not a set of parameters, it's a set of models. And the models can be simulated, not in the CPU, but in the GPU. So we have GPU simulators that can simulate these models in parallel. And then the output is the fitness of the model that goes back to the CPU for the CPU to rank them, keep the best ones, and then combine them. So in this crossover and mutation, what's happening is that models, equations from the different models are merged. So maybe some part of the equation is coming from this model, other part from this other model. Parameters are coming together. Mutations can include new equations in the sense of new genes in the network, can remove genes in the network, etc. So this methodology can discover the number of genes, the network topology, and all the parameters needed to build these partial differential equations to create the spatial patterns from input signals. The speed up of this GPU implementation is 700-fold, so we are very happy about the speed up of this methodology compared to a single CPU. And then the second problem is how do we quantify how good a gene regulatory mechanism is? So one challenge is that sharp edges are very hard, especially corners are very hard to do synthetically, because you need a lot of regulation, very, very strong regulation to create a very sharp edge. So we introduce tolerance parameters. So the idea is that we are blurring with a kernel convolution method, we are blurring the patterns to remove those sharp edges, to be able to compare them more linearly. And that can be adjusted as you want. So the fitness function, the error of a model depends on the shape, which is the shape of the pattern that has been is the input, and the one that is developed through simulation, both through the blur kernel, and then an equilibrium penalty, because we want patterns that are stable. We don't want dynamic patterns that change. We want patterns that when we simulate them, gets into the steady state. So we have a couple of parameters. One is the size of the kernel, which makes the pattern more or less blurred, and then also the threshold between the concentration in the simulation and the concentration of the pattern. Most of the pattern I'm going to show you looks very binary, but the system is continuous. We can also apply it to gradients, etc. So how does it look like when we run an evolutionary algorithm? So say that your goal is to produce this triangle. So in about 10 hours of computation, at the beginning, the error is very high, right? These are basically random models. So there's a connection between the green and the red gradients, and the output is going to be the blue. The output is like a reporter gene to make the pattern, the spatial pattern. So the beginning, as I said, the models are very bad, so the error is very high. We are up here, but as time progresses, this is real time. The models get, by chance, gets a little bit better. So they are selected. You can see there's a shadow here that is slightly like a triangle, maybe a blob. Now the blob gets a little bit stronger, so the model gets a little bit better. The computer continues modifying the model through this evolutionary computation. It gets like a semicircle, which is better than a blob. And then by generation 3000, it's almost a triangle. And by generation 4600, we get a perfect triangle that recapitulates the input pattern. And it is the gene regulatory mechanism that has been discovered and includes all the equations, the parameters, etc. We can simulate these models in the computer. So this is the simulation of a triangle pattern. And the cool thing is that these are the inputs and the output genes, and these are the intermediate genes, all these guys together. And the mechanisms that we observe that evolved through this machine learning optimization looks like developmental biology. So we can have many different patterns: square, circles, triangle, diamonds. And look at this. This is A, is an intermediate that is creating this domain that makes these two sharp edges. And then this one comes from the negative regulation of the green and the red morphogens. So as I said, these are very developmental biology-like patterns. And we can, as I said, we can tolerate we can change the tolerance of the method with those kernel sizes to make the pattern to be more approximate, like here. The first column on the left, the patterns are more tolerant, so they are more approximate. You can see that the circle is not very circlish, and the diamond is slightly off, but the mechanism that can create these approximate patterns are much simpler, right? On the right is the more complex patterns. So uh, lowering the tolerance of the method can return much more complex gene regulatory mechanisms, but the patterns are much more closer to the target. Of course, it takes longer. So on the right, you have the time that it takes to produce these gene regulatory mechanisms. And it runs from 1 hour to 30 hours in a big, in a medium computer. So here are some more simulations with more complex patterns. We can, as I said, basically create any patterns. So we have hearts, we have smiley faces. We can have the logo of the university I put at the beginning. We can have letters. So almost any pattern can be created by this methodology, including gradients, etc. So now the next question is, okay, this is all in the computer, right? Can we engineer them? Can we engineer the patterns and the networks that we have designed automatically? So this is still very work in progress, but I'm going to show you a preview. So the idea is that we have a target pattern that we want to engineer for any application. It can be biomedical, it can be maybe for producing some kind of tissue shape, or producing something for engineering applications, bioengineering. So the idea is you have a particular pattern. That's the input for the machine learning. The machine learning designs the gene regulatory network that, when simulated, produces the pattern. And here is the simulation. Very good. So the idea is, can we engineer circuits genetically, plasmids, that when we transform cells with these circuits can react to these input signals to produce the pattern? Again, this is a very early work, but this is a petri dish that we have engineered, where we have a field of cells engineered with the circuit that was designed, and the patterns, the L pattern, is shown in the petri dish, right? So again, this is a very early work, but we have promising results of engineering these patterns. Are we going to make the smiley face sometime? Maybe, we will see. Right. So in the last minutes, I want to just touch very fast on the human health. Right. So we apply also this methodology to try to understand cancer progression and other diseases. So this is, we have collaborations with the University of Maryland Cancer Center. It's a comprehensive cancer center assigned by the NIH. And the idea is that we have progression data for 467 patients. And this progression clinical data includes, of course, patient data, age, death, BMI, leukemia parameters, including the blast counts, cytogenetics, genetic mutations for genetic panels that we are performed to these patients, and then, importantly, what treatments each patient was receiving, right? Including the chemotherapy and induction chemotherapy, more experimental treatments. First, we did the PCA analysis to see if there's anything that is very clear, right? Of course, there's nothing that is very clear. There are the data is very complex. Otherwise, the doctors wouldn't have a job, right? There's many patients that are very complicated that we don't know why that happens. If we plot the data dynamically, this is how it looks like. So we have lots of type of heterogenic data of the cancer progression and also the treatments that were performed to the different patients, and then how the disease progresses in terms of number of blasts. So the idea that we are performing, this is still unpublished, is to try to introduce these dynamic data sets into machine learning, very similar to the ones I provided before, to create dynamic models that now can be used to predict what is the best treatment progression for a particular patient. The models look complicated, but this is an example of how a model can integrate very, very heterogenic signaling mechanisms to predict the progression of the disease according to the different treatments that the patients were receiving. The idea is if we get a model that is good, that can predict our data sets, including training and testing, can we use that to inform medical doctors of what is the best treatment for patients, especially for those that are not responding well to regular therapies. Those are the ones that the medical doctors need help with. The idea is that we we don't have time, right, to test different treatments to a single patient because they're dying. So the idea is, can we give them at least the best shot according to these machine learning methodologies? That's kind of the goal of this project. That's my last slides. So in summary, my lab is doing systems biology of growth and form. We mix different type of signaling mechanisms, including genes, metabolites, and biophysics to try to understand development and regeneration, create synthetic biology networks that can be implemented, engineered in vivo, and then cure human health diseases. I want to thank my lab, all the funding sources, all of you for coming here and listening to my talk. Thank you.
Questions for Daniel?
Yeah, I thought that was pretty neat. What would it take for your model to be able to incorporate like changes in the stages of life, like sudden metamorphosis or turning from a triangle into a heart?
Oh, that's a great question, right? So you want a more of a dynamic model, right? Instead of having a single output, you want to have a dynamic output, very similar to what the worm is doing. So that's not hard at all. We just need a fitness function that instead of checking only the last time step, that's how we have it set it up now. The last time step needs to be stable and needs to match the pattern. We can have a middle one that needs to match the pattern, but it's not stable, and then a final one, right? That is the second pattern that you want to do. Yeah, we could do that. It would be interesting to also see the difference between the networks, right? Are they a mixed network? Are they reusing parts of the network to produce a mixed pattern?
In your pattern formation experiments, is what every cell is doing, is it just dependent on the concentration, or does it also take input from the neighboring cells?
Right now, only the concentration. That's also work in progress to try to introduce communication between the cells. Can we create Turing patterns? That would be great, right?
Does the model also take into account epigenetic mechanisms, like the one you showed with cancer?
No. Yes.
Could you
We have the data. Right. So the data, the question was, can the model take into, or does the model take into account epigenetic mechanisms, especially in the cancer project? And I said, we don't have data about that. So the answer is no. Can we introduce them? Absolutely. Yeah.
I have a kind of dumb question.
Oh, there's no dumb questions.
So have you tried to cut the worms vertically instead of horizontally? So to stay serious.
We can cut the worm anyway. Vertically, it's going to regenerate the second half. I mean, each half is going to regenerate the second half. Diagonally, the same. Any cut that you can think of. The only thing that doesn't regenerate is the pharynx. The pharynx is the organ that they used for eating. There's no stem cells in there. So you need the stem cells to produce the rest of the cells. And also above the eyes, anterior to the eyes, the worm cannot regenerate either because there's no neoblasts there. Why? I'm not sure. I think it's because of the brain is interfering for the neoblast migration. But they cannot regenerate anything above the eyes. You cut this piece, right? You're going to regenerate the piece, but the piece that is coming from here, from the anterior of the eyes, does not regenerate a complete worm. There's also very cool experiments where we can irradiate the worm with X-rays to delete to kill all the neoblasts, all the stem cells in the body. And the worm, with time, dies because it needs to replace all the cells in their bodies. But we can take a single neoblast from another worm, a single stem cell that is pluripotent, inject it into the worm that has been irradiated, and the worm lives forever.
The back.
Yeah, so in your simulations, do you apply any selective pressure for simpler models?
We do. We do it is we have mutation operators, right? And there are mutation operators that can increase the number of genes, right? You can introduce a gene into the model, and that makes the model more complex. And the same, we have mutation operators that can introduce new interactions between the genes. And then we have mutation operators that do the opposite. You can remove completely a gene from the model and remove links from the model. So what we do is that the mutation operators that can make the model more complex have lower probability than mutation operators that can make the model simpler. And without touching the fitness function, that gives you enough evolutionary pressure to try to make the models simpler. Another trick that we do is that once the model reaches zero error, according to our thresholds, we don't stop the algorithm there. We keep it running because that model in general is complex. But with evolutionary time, it can't simplify because, again, there's a bias towards simpler models. So we keep the evolutionary algorithm running until we don't see a simpler model after, I don't know, 1,000 generations, I think it is. So that simplifies the model at the end as well. So those two methodologies we use to try to return simpler models.
Okay.
So when you talked about development and by implication evolution of development, and then you also talked about synthetic biology. In a way, I think those sort of work in opposite directions. So when you're doing synthetic biology, you say, I want this shape, and then you say, can I, come up with a set of differential equations that will give you that shape.
Right.
So but evolution doesn't do that.
It doesn't?
How I would pose the question is, if we see similar patterns over and over again, and then underlying circuitry underneath that, is that there's only a certain number of reasonable solutions to a problem, and so evolution finds them over and over? Do you think
that's the case? I believe that. Right. So the idea is evolution is a tinkerer, right? Natural evolution is an implementation, you can think of evolutionary algorithm. Right? Evolutionary algorithm is the abstraction of the methodology. Biological evolution is very complex, of course. We don't know what are the fitness, and there's a lot of organisms interacting with each other, right? It's extremely hard, but it's the same concept. The organisms are evolving towards a goal, which is better reproduction. Do we see convergent evolution in the networks? Can go back to 10 years ago, right, where the concept of motifs were developed by Uri Alon. So they found feed-forward networks. I mean, it's a very small motif, right? But they found feed-forward networks being everywhere in the biological systems and human-made systems, right? So it seems that, yes, there's a convergent evolution towards particular mechanisms to solve particular problems.
Can I do devil's advocate for a minute?
Sure.
So Eric just asked, is there an an advantage to having simpler models, and it sounds like you built that in, but
Mhm.
not on a fitness function level.
No.
But if a circuitry evolves that does the job and it's not that simple.
Right.
Will it get stuck in a local minimum and be perfectly happy with that, or?
It's possible. There are research, right? Also trying to see metabolic networks, if they are optimal or not. They have studied metabolic networks across many different species of bacteria.
Mhm.
And they show to be in the Pareto front, right? They can control between different parameters, right? And they show how those metabolic networks are optimizing, are very, very highly optimized. So yeah, there can be different minima, and probably not everyone is in the totally optimal one, but I'm sure that they are getting into those minima.